Thank you so much, Patrick. Hi, everyone. A very warm May welcome to our new attendees and welcome back to our regulars. I'm thrilled to see so many of you logging on. My name is Marissa Eastman, and I work on the side of the Catalyze Coordinating Center, leading our skills development offerings and programmatic outreach. So you'll recognize me from this webinar series. We host a monthly webinar, typically every month, as well as an annual meeting, and do various programmatic outreach as well. So that's all under my purview. Today's topic is one that generally garners a lot of interest, so I'm very happy to see so many of you, and that is Catalyze funding opportunities. We scheduled this webinar very intentionally as our next product definition application receipt date is coming up on June 18th. So today we'll have two members of Catalyze leadership speaking about the program, the current funding mechanisms, and how to apply. There will be ample time for question and answer, as Patrick said, so please utilize that Q&A function to submit your questions throughout the webinar. And at this time, I'm very happy to pass the microphone over to Dr. Diana Severynse-Stevens, who is the director of the Catalyze Coordinating Center.
Thank you, Marissa. Can we move to the first slide? So today, Mike and I are going to provide an overview of the Catalyze program with a focus on the current funding mechanisms. Then we'll talk a little bit about the updated product definition notices of funding opportunities. And as we've said, there'll be plenty of opportunity for us to address any questions. Next slide. So the Catalyze program offers investigators funding, preclinical services, technical support, and training. The two key components to Catalyze are the product definition component and the preclinical services component. And I think it's really important in the focus of today to help everyone understand what the differences are. The product definition component awards grants that can be up to $1 million in direct costs for up to 3 years. They can be R61 transitioning to R33 grant proposals, or they can be direct to R33. These grant proposals go through the NIH review system, and there are opportunities for therapeutics, devices, and enabling technologies or transformative platforms. And Mike will provide more information about the specifics related to those grant proposal requirements. There's typically 3 application cycles a year. We had one in February. The next deadline is June 18th. And then there's also a deadline in October. So for the product definition component, investigators apply for grants and the funding is received directly or is awarded directly to the investigator and their institution or company. On the preclinical services component, it's very different. You aren't awarded funds. No funds go directly to a PI or to their institution or their company. It is preclinical services, so you can request specific types of activities that would support a preclinical development program. So you may be in need of manufacturing or toxicology studies. You can request those services, and the funding will be provided directly to the CRO or the CMO, so the contract research organization or the contract manufacturing facility, and that's managed through the coordinating center. And typically we have 2 application cycles a year. We're in the middle of our 2nd application cycle. For this fiscal year, we don't currently know what the application deadlines will be for the next fiscal year, but you can check our website and we'll make sure to update through newsletters and other announcements once we know what those will be. So again, the focus is for preclinical services to provide gap-filling studies through the funding of activities at CROs and CMOs. Next slide. So again, we have the product definition and preclinical components, and often folks aren't sure, where they belong in Catalyze and what kinds of activities are funded or provided services are provided through these different components. So this figure highlights some of the types of activities that occur within the product definition versus the preclinical services component. On the product definition, and this is to the left with the lighter blue boxes, for therapeutics, there may be activities funded that would be lead optimization, cell line development, ADME PK studies, in vivo efficacy studies, early drug substance characterization, and some drug substance stability. That would be on the therapeutic side. So for small molecules, biologics, cell and gene therapies. Within the device and diagnostics and enabling technology platforms, there may be early stage activities related to prototype design, prototype testing, bench testing, characterization, and maybe some early feasibility studies. Those are the types of activities that you would be requesting grant funds for under the opportunities for devices and enabling technologies. Under preclinical, the types of activities that would typically be supported through preclinical services for therapeutics would be the development and validation of bioanalytical methods, analytical methods, scaling up of manufacturing, transfer of manufacturing from, say, a laboratory or small company to a CMO. Drug product formulation and stability, toxicology studies, safety pharmacology, all of those types of activities would typically fall within preclinical. On the enabling technologies and the device perspective component for preclinical services, there may be additional feasibility testing, verification validation testing, observational studies, and other types of things that would fall within that preclinical space, preparing you for a submission to FDA. Next slide. Now, one aspect of Catalyze that's a little unique is that not only do you receive either product definition funding or preclinical services, there is the availability of free resources within the coordinating center. And what I mean by free resources is there are a very diverse group of products in the portfolio, but we have experts and resources available to all investigators in the program through the coordinating center and a network of consultants. And because the portfolio includes small molecules, biologics, cell and gene therapies, devices, in vitro diagnostics, enabling technology platforms, we try to have experts that sort of are across the spectrum of very diverse product types. And so we have medicinal chemists, toxicologists, pharmacologists. We have folks that are bioengineers that can help in prototype design, regulatory affairs folks that can help you engage with CDER, CBER, or CDRH. And we also have commercialization support. So all of these resources are available to you when you're in the program. Next slide. We're often asked, how does my product fit in Catalyze? And we've kind of walked through the product definition and the preclinical services activities that typically occur when you are either receiving a grant or preclinical services. But we also developed this decision tree to help an investigator sort of walk through where they are with their product development and how they fit into the Catalyze program. So I think the key question here at the top is, have you identified your product? And do you know how you're going to use it? What is its indication? If the answer to that question is no, then I think the next thing to look at are what are the prerequisite activities required to get into the Catalyze program. On the device side, you need to really at least have an early concept, an understanding of what your platform is going to look like, what your device is going to be, the basic concept of your in vitro diagnostic. If you're not quite there, if you haven't started like early prototype development, then you may not be ready for the Catalyze program, even within the product definition component. On the therapeutic side, you should have confirmed your mechanism of action. You should have some in vivo proof of concept testing Again, if you haven't done these types of studies, if you haven't generated this data, then you may not be ready for Catalyze, and you may want to pursue an R01 or other types of funding. If your answer to this is yes, we have this very early data and we have some information that justifies moving forward into product definition, then I think the key thing is, what are you looking to do with the funds that are provided through Catalyze? Typical activities under a product definition grant award for a device or enabling technology would be, you're working on prototype design, you're doing early feasibility testing, maybe animal testing, if that's something that's appropriate or applicable. And you may also even begin some verification validation testing. Those are the types of activities that typically occur within the product definition funding component. On the therapeutic side, you may be requesting funds to do lead optimization studies, to do additional in vivo efficacy studies, to start to optimize your synthetic process, to begin non-GMP manufacturing, maybe some dose range finding studies. These are the types of activities that investigators typically perform under product definition with product definition funding. So these are the key activities. If this aligns with where you are in your product development, then product definition grant application is probably appropriate. If to the original question, have you identified your product and its indication? If your answer is yes, then you may be ready for preclinical services. Again, there is some overlap between the two, but trying to define very clearly here the types of activities that go on in the two different components will hopefully help you determine which component of Catalyze best fits your project. The prerequisite activities to be considered for Catalyze preclinical services on the device side would include prototype design lock. You've decided what your final design is, or you're very close to it. You've already begun some validation and verification testing. You are ready to start GMP manufacturing, or you've already started some of that work, and you have a clear development or regulatory path that may have already engaged with the FDA. On the therapeutic side, your lead compound or cell line is well characterized. You demonstrated in vivo efficacy. You're looking to either engage with FDA in a pre-IND meeting, or maybe you already have. You have a clear development of regulatory plan. And in both cases, to apply for regulatory services, for preclinical services, you will need to have not only engaged with FDA if you're requesting GLP or GMP activities, but you will also need to have vendor quotes for services at the time you apply that need to be relatively recent. So if, for example, you say, well, we would like to do GMP manufacturing, but we haven't had a pre-IND meeting yet, it may be that you can apply for regulatory only services first and then go back and apply for preclinical services. I'll talk about that in a minute, and that's a new service that we launched in the fall. So if you have the data from these types of prerequisite studies and activities, then preclinical services may be appropriate for you. If you are applying for preclinical services, this list here in the boxes at the bottom to the far right give you examples of the types of activities that we typically in preclinical services support. On the device side, that might be moving the prototype to GMP manufacturing, sometimes larger animal testing, observational studies, and then help with the preparation of, say, an IDE or a 510K. If you're in the therapeutic space and you're requesting preclinical support, then you may be requesting dose range finding studies or GLP toxicology studies, again, assuming that you've had a pre-IND meeting. You could be requesting non-GMP manufacturing or GMP manufacturing, or you could be requesting support with a pre-IND meeting along with some of the other services or preparation and submission of an IND. So in the case of product definition, you'd be submitting a proposal through grants.gov. In the case that you fit best in preclinical services, you would be submitting an online application through catalyze.org. Next slide. So as I mentioned, we have a new service that's available. It's standalone regulatory only service. We launched it back in the fall. Anyone can apply if they're not in the program. If you're already in the program, regulatory services and support are available to you. But if you're not in the program and would like to get Catalyze support for regulatory activities, including consultation, regulatory strategy help, preparing for a meeting with FDA, assembling documents or submitting documents to FDA, we can do all of those. And it's a simple process. There's an expression of interest form similar to the preclinical services application, expression of interest form. And then you're invited to submit an application. The one key eligibility criteria is that you need to be, within TRL levels 1 through 6. You need to be a US-based academic nonprofit or US-owned for-profit institution. Next slide. So in just wrapping up the preclinical and regulatory support portion, the key thing for this is to understand the differences in what you do for preclinical services, product definition, and regulatory support. Again, preclinical and regulatory support are similar in that you submit first expression of interest form online, and then you'll be invited to submit a full application, assuming you fall within the scope of Catalyze and within the high priority areas for NHLBI. And you can download the full applications for preclinical and regulatory by going to nhlbicatalyze.org, these two links. We will be providing the slides after the presentation through the website. They'll be probably a few days, maybe early next week, they'll be available so that you can look at some of the details that may have been harder to capture, particularly in the decision tree during a short presentation. So at this point, I will hand it off to Mike.
Thanks, Diana, and thanks again for joining. So, and also to remind you that in addition to the slides, we have a very detailed website listed here and a curated series of FAQs related to both the product definition program, which I'll cover, as well as the preclinical program that Diana discussed. Next slide, please. Okay, so I'm going to walk through the funding opportunities related to the earlier stage of the pipeline, the product definition program, and kind of discuss some of the the key updates since we renewed the program, as well as the differences between the 5 related funding opportunities. So next slide. All right, so walking through these, we have a standalone enabling technology transformative platform, R33 mechanism for 2 years. And then for the devices and the small molecules and therapeutics, we have related biphasic mechanisms for earlier stage projects, as well as a standalone R33 opportunity for kind of some later stage projects. Next slide. And regardless of which program you come in through, all the 5 NOFOs or funding opportunities have the same special requirements, and these are special relative to your average NIH, R01 or other type of R mechanism. And these are listed here on the right, and they include project management, both the adoption of project management, but then also to identify an individual who will serve as your project manager, preferably not the primary principal investigator. For each of the specific games that you propose, we expect additional milestones and a timeline to help us and you track your progress toward those specific games. There is a focus on IP and regulatory strategy, and for the 4 NOFOs, except for the enabling technology, this is a separate attachment that you must include, otherwise your application will not be renewed or reviewed rather. And then there's a focus on the NIH is the definition of rigor and reproducibility. But then we also take another lens, given that these are usually projects that go in front of the FDA. So if there's an additional rigor and reproducibility that the FDA requires, those are things that we look for. And then for the R33 projects or applications, we have an expectation that you'll go out and try to find non-federal funds to help advance the technology, as well as identify an accelerator partner to help you develop the network of business and supporters in your regional network. Next slide. So the first and standalone project is the Enabling Technology and Transformative Platform. And really this is for technologies that have had the feasibility already worked out, and that'll be expected as preliminary data. And really the idea is that these are projects that are new platform technologies or transformative platforms that, if developed, will really relieve a bottleneck or advance heart, lung, blood, sleep research and therapeutics. And in general, these types of technologies do not have a specific indication at this point, given that they are platform technology. And in addition, we're looking for really new and novel approaches to some of these projects and challenges. So again, this is a R33 mechanism. And if you're familiar with other grant funding, a lot of the R21 type of projects here at the NHLBI are appropriate for follow-on funding to the R33. However, there is no requirement for prior funding for any of the Catalyze projects. This specific program is for 2 years of funding, and the direct costs were increased this last round to $350,000 per year. And so from there, we will move on to the therapeutic suite of projects. Next slide, please. So the small molecules, biologics, and new to this renewal combination products that have a drug component should be submitted to one of these two mechanisms. On the left is the R61, R33 biphasic mechanism for early stage projects. So in here, you can think about trying to get to that lead series or lead optimization. So we receive a lot of applications for screening library compounds for lead series type of applications, and then the R33 for those is to validate their lead series in some sort of animal model to get those preliminary feasibility studies. So it's set up with a lot of flexibility. So if you have a fairly early stage project, you can propose to do 24 months in the R61 or phase 1 of the program, and then have some key no-go criteria to advance to the R33, where you would complete the third year of funding, and vice versa. If you already have a lead compound or lead a few hits, and you want to just do some additional validation work, you can propose 1 year in the R61, and then transition to the R33 to support some kind of larger animal studies if you're at that stage. At the time of application for the R61, R33, you are not required to provide any supporting evidence of an accelerated partner or matching funds. However, if you do have matching funds at that stage, you should provide some sort of evidence. This is not peer reviewed. However, programmatically, this does go into some consideration, just kind of as a flag or indicator that there is some sort of market interest in the technology, even at this early stage of application. If you have a slightly later stage technology, we have the R33, direct to R33 option for both the therapeutics, the 018 for this series of NOFOs. And again, this provides 2 years of funding with the increased direct cost of $400,000 per year. And at the direct to R33 application stage, you must provide a letter of support from the accelerator partner, and you must provide some sort of evidence of a non-federal match. We recommend 25%. However, we do realize that this is a difficult time to be raising funds, and depending on where you are geographically, it may be easier or more difficult to identify and raise funds. So review the notice of the NOFO instructions for providing that supporting letter and evidence of matching support. Next slide. And set up very similarly for medical device and diagnostics. So the early stage prototyping, work is best supported in the R61, R33 on notice NOFO here at 019. Again, you can propose to do the 2 years if you have several components and you haven't quite made that first prototype yet, and that'll allow you time to prototype each of the different components and get them all together for that first prototype before some sort of validation step in the R33. The only difference between this and the other series of notice NOFOs is the difference in the direct cost being slightly less, knowing that most medical device and diagnostic development is slightly cheaper than therapeutics. But the same requirements and expectations regarding the accelerator partner and non-federal funds hold true for this series as well as the prior biologics and therapeutics. Next slide, please. So in summary of everything, just this simple diagram of what we support. So your basic R01 research, if you have some new emerging exciting technology that you want to validate, or an R35 for that matter, the product definition R61, R33 would most likely be the right avenue for you to pursue. And as you have more and more mature technology, even after a phase 1 SBR or STTR, the R33 funding mechanisms may be the most appropriate, shown here in 3 and a little bit of 4. But as Diana mentioned, if you have sufficient data to support a pre-IND meeting with the FDA, you may not have had it yet. The preclinical services may be the most appropriate avenue for you to explore. And key to all of this, and on the next slide, I think we can kind of end the formal presentation and start to, oh, sorry, I have one more slide. So we just wanted to give you some examples of funded projects for each of the 5 NOFOs. So going at the top, we have a biphasic R61, R33 just recently funded. And this is a gene therapy focused project where they're doing some of the final validations of the AAV and gene product kind of delivery. And then they'll transition into the R33 where they'll do some preliminary animal studies. Slightly more mature in that second one is a genetically engineered pig heart as a bridge to allo transplant. So this was actually coming in for enabling tech transformer platforms. And likewise with the device components, we have a machine learning and device and hardware type of project that they still have to prototype the 2 components working together. And the R33 example for the device is a sealant where the investigator has done significant amount of de-risking and preliminary work to demonstrate that it's feasible. And now they're just doing some final studies in a large animal model in preparation for FDA approval. And then here below, the last 2 rows are 2 examples of enabling technologies that are kind of illustrative of the NOFO itself. And all of our projects are listed on our website. These link out to the NIH reporter so you can get the grant number, a little synopsis of what they're doing, as well as the institution that the investigator belongs to. With that, we can go to the FAQs. And I also, at this point, want to introduce my colleagues, Nitin Agarwal from the Blood Division and Rahul Thakur from the Division of Cardiovascular Sciences, who are also part of the Catalyze core team. And the reason they're here is to, again, remind all the applicants to contact either the Coordinating Center or the NHLBI to discuss your project before you submit it. We know that if you speak to us first, you have a better chance of getting a fundable score. And it helps both us know what's coming in the door and also helps you understand how best to position your project for the various parts of the Catalyze program, or Rahul and Nitin can also help you identify additional NIH funding opportunities that may be outside of the Catalyze program. So at this time we have, we're going to address these pre-submitted questions. And maybe Nitin, since you are a former NIH R01 awardee, maybe you can walk through the differences between the first question of an R01 and SBIR and where Catalyze might fit in.
Sure. So hello, everyone. I'm a program director at NHLBI, and I'm leading the Catalyze program also from the Blood Division. So the first question is, can you provide information about this grant versus the traditional R01 or SBIR with regards to requirements, review process, and funds for accelerator partners? So an R01 is, as you all know, is a basic research grant which involves fundamental research on a specific topic. It could be the development of tools and technologies as well. However, it's a more elaborate and a more extensive program, not geared towards commercialization. SBIR, on the other hand, is mostly geared towards commercialization, and your technology should be matured enough so that you feel confident that it is ready for commercialization. And that is when you apply for the SBIR, where you would be doing most of the commercialization work, most of the legwork. What segregates or what separates the Catalyze program from the other two is the quality, is the amount of services that you receive in the Catalyze program, as Diana and Mike has mentioned previously, that you will receive thorough support in the areas wherever you need support. For example, the FDA approval process, or the regulatory processes, or the toxicology process, or communication with or finding specific vendors for developing or GMP facilities. You will receive support for a variety of these aspects from our experts from the coordinating center. So that is the primary difference between the Catalyze and the SBIR, that in SBIR, you will be doing most of the work on your own, and it is a more matured technology for commercialization, whereas in Catalyze, it's a biphasic program.
Thanks, Nitin. And just two other points to point out. So the SBIR and STTR program have very specific percent effort requirements that the Catalyze program does not. And in addition, the R01 is more hypothesis driven versus this being more product definition. So I think I'll address the second one, which is a question related to which institutions should apply, academic versus startup. There is no difference from the peer review group or from the programmatic review. I think it should be whoever or whichever institute is the most appropriate to support the work that you're proposing. And again, we have no it doesn't impact your review at all. So maybe Rahul, I can ask you to address the third question of how to improve your application submission for a resubmission.
There's a couple of options. Thank you, Mike. One is prayer and incense. What's more practical is to consult with your program officer, reach out to your program officer, myself, Nitin. We're all here to help you. We're here to help try to, I guess, decipher sometimes what a summary statement says within the context of our roles. And to be honest, Mike alluded to it momentarily a few moments ago. Consult with us before you submit. There are so many issues or angles that we can provide you guidance on. We can anticipate versus react. It's significantly easier to do that and come in with that A0 that way. And you put your best foot forward. And if you happen to not get funded, hopefully you're discussed, and then we can work with you again to improve the score. You have to keep in mind, as program officers, we're in effect your advocates, your champions, in many cases your SMEs on the program side here. We want to see you succeed. We want to see you go forward, but it requires knowledge of what you're doing, especially before it goes to review, for us to maximize our effort with you.
Just to add, Rahul, I was recently talking to a company who had applied for the R61, and they had no idea. This was the first time they were applying for an NIH funding, so they had no idea how the process works. And so that is where we come into the play, and we can guide you through the process. But we highly encourage you to first go through the website. Every single detail is given on the NIH website, and after that, if you have additional questions, then we are here to help you.
Yeah, and again, some of these questions I've seen are actually in our FAQs on the website. So again, we're happy to go from here, but that more thorough list of these questions that we see, as it says, frequently are addressed there. So the fourth question is a private company being an accelerator partner, and what counts toward match support. So just in general, the accelerator partner, the rationale for that requirement is to ensure that primarily academic investigators are reaching out and exploring other entities in their, their networks, really with a focus, the accelerator partner, on helping connect and develop the business components that are essential for advancing these technologies out of the academic research and into a commercial pipeline. So we have several ways that we can address this. So for academic partners, most universities now have some sort of innovation zone or innovation office that can assist you either as the accelerator partner or identify or know accelerators within the university's network. A private company, generally, they have a CEO, and if they're mature enough, we are okay with the CEO indicating that they would be the accelerator partner. Sometimes CROs have a venture arm that is a really awesome accelerator partner because they can help you kind of scan and identify the full development pathway to marketplace. So there's a lot of flexibility with who the accelerator partner can be, and generally should be someone or some entity that can help support the business development side of the technology advancement. And then what counts toward match support? So as clearly written in the funding opportunities and on the website and reviewed here again, any non-federal dollar amounts can be counted toward match support. This includes, for academic investigators, a letter from the dean indicating that the university will cover salary for the duration of the grant, or any dollar amount related to the 25% of the R33. Ideally, it is actually cash that can help accelerate the development and do other types of activities related to the R61 and or R33 activities. Number 5 is related to number 2, and we covered that. So small businesses, large businesses, nonprofits, any US-based entity is allowed to apply. And that brings up another late-breaking notice from the NIH that was recently submitted this week, that foreign subawards are no longer allowed until further notice for all applications. So if you currently have a sub-awardee who is a foreign component or institution, you will need to change that, or it will not be reviewed on June 18th. I'm going to ask Rahul to take number 6 regarding submitting an SBIR and Catalyze at the same time.
So again, not to be glib, just because you can do something doesn't mean you should do something. There's a very strong possibility both of these types of applications would go to different study sections, which is great. If you are going to pursue this sort of strategy, you need to make sure that the scopes of work are entirely independent. Any risk of overlap in this sort of situation, you run the risk of losing one or both, if they're both meritorious, that is. The key thing is independent scopes of work. Please consider the FAQs and all the information that we have before you, and meet with your program officer, especially if you're going to pursue this sort of strategy. Ideally, it would make sense to choose one project in one cycle and take the next one to the next cycle, depending on how mature your prototype, your project, your product, whatever is. It allows us a chance to kind of spread our investments over different types of companies, different PIs, different groups, different technologies. So again, you could do it in the same cycle. I'm not sure it's always advisable.
Thanks, Rahul. And maybe, Nitin, can you address the second question related to the applicant or the PI?
If an SBIR grant was awarded to a PI within small business, is it possible for the same PI? Oh, this one, Rahul covered the 7th, right?
He covered the 6th one about applying in the same cycle. So maybe just addressing a PI listed on both an SBIR and then also a Catalyze.
Yes, so The same PI can apply for both grants, but as Rahul mentioned, it is critical that your scope of work for both grants should be different, and we encourage you to apply in different cycles for a better success possibility.
Right, and maybe just related, we have had active SBIR awardees who are the PI successfully apply in a different cycle and have a Catalyze award at the same time. So it's possible. Again, you just have to demonstrate that you have the capacity to oversee both projects. Diana, maybe I can ask you to address number 8 about getting a product through final design and engineering, and maybe explain or describe some of the resources available at the coordinating center.
Sure. So obviously it depends on which component your product is in. Is it in a product definition phase where you're getting funding and you're doing the majority of the work, within your laboratory or company? All the resources that I mentioned are still available to product definition projects. So we do have some bioengineers. Their engagement with the product definition projects is essentially more as consulting. They both have extensive regulatory experience as well, so they can provide both engineering guidance and input, as well as regulatory strategy and just general planning in terms of when and how you should approach FDA, and can help with that process. So if you have your own funds through the product definition, then it's consultation on the engineering and the regulatory affairs side. If you're in preclinical services, again, you have the same resources available to you, but the bioengineer will be a little more engaged if there's prototype manufacturing going on. That individual would be engaged in all of the contract sort of meetings, or meetings with the contractors, the vendors doing the work. Of course, the PI is also engaged in their team in all of those meetings, but it's a little more hands-on. In a product definition project, typically there's quarterly meetings, although there can be technical meetings in between those quarterly meetings. In the preclinical services component, there may be weekly or monthly meetings to help with the vendor activities going on in support of the engineering and design work.
All right, and then the last pre-submitted question, I think I'll just touch on again. So how do you identify accelerator partners? So if you're at the R33 phase and you're at a university, that would be the best place to start, starting the tech transfer office, or look at kind of, bio.org lists them. There's several ways to go about it. A web search is probably the best. And then IP arrangements are typical. There is no typical, because this program is so broad. So for some of our drug development, there is no IP because they haven't identified the molecules of interest, all the way up to they have several issued patents and are in agreements with out licensing or in licensing technology. So the point is not to review the level of IP arrangement that you have. The point of the requirement is to communicate to the applicants that we expect that these are commercially viable products or projects that will ultimately have IP. So we want you thinking early on how you can protect that IP to make the technology commercially successful. So I forgot to mention one other update with the renewal of the NOFOs, and that is for the small molecule therapeutic R33 and the device diagnostic R33, but not the enabling technology. We now allow for renewal applications, which means that once you complete your R33, that if you're successful, you may reapply to the program with new milestones as a renewal application. So if you're a current awardee or have recently graduated from the program, please reach out to the coordinating center or us, and we can discuss the mechanics of it. But in general, the NIH website has information on renewal type applications, and the Catalyze program follows those general guidelines. And I'm seeing, let's see, do we have any other questions, Marissa, that we need to call out there?
I see a question from Jay Potts. Can you explain the difference between the aims versus milestones that are required in Catalyze versus the other R programs? And even though the answer was posted, but I'll elaborate on that a little bit, that for the R grants, the aims that you mentioned could be broad aims and could require a lot of time to accomplish. But as it was mentioned previously, for the Catalyze, your milestones should be highly focused and should include adequate deliverables. And Catalyze grantees are required to meet quarterly with our team. And in each of those quarterly meetings, you will be required to present the achieved milestones that were defined for that particular period.
Thanks, Nitin.
Mike, there is a question regarding the R33 renewal, that if a current investigator with the active R33, can they apply for preclinical support and also an R33 renewal?
So the rationale for now allowing the renewal application is the observation that many of our investigators were not completing the R33 program ready to compete for the preclinical program, as in they hadn't sufficiently developed their technology to get to that step just before FDA review, as described by the preclinical requirements. So this additional 2 years of R33 funding should help close that gap between where some of these projects end at the R33 stage and where we expect them to be prior to applying for preclinical services. So for specific projects, it's best to just kind of meet with us either in a technical meeting or quarterly meeting to discuss your options for that renewal submission versus preclinical program submission.
Okay, thank you.
And then I just want to address some other reoccurring questions that they're on the FAQ page, but we'll address them here. So the SBIR program has specific guidelines on how much work you can outsource to either another subaward or to a CRO or such. The Catalyze program does not have any of those requirements, so you should propose to do the work wherever is the best place for it to get completed efficiently and economically.
There is another question. If we have the option to apply either via our academic center or spin off a company, which would be the better path?
So we address that in number 2 here. There is no preference from either the review side or the programmatic side.
Okay, thanks.
So just a reminder that we do have a quarterly newsletter as well as some other announcements, and there's an option to sign up for these alerts through the website that Marissa is showing you here. That will help you know when we are opening or closing different programs. So just to address another question we're getting a lot, we don't have guidance yet on when the preclinical program will open back up for applications. And so the best way to know about that is to, again, sign up for our newsletters. We don't send a lot. So you should see them when they come in.
Awesome.
I would like to emphasize that you would be surprised how much information is available on these websites and in these newsletters. A lot of planning and effort goes behind developing these and sending these out. So please, you are encouraged to go through those first, and most likely most of your questions will be answered from there. And then we are here to provide additional information.
And then a question about indirect costs. So follow a moving target, I guess, for the last several months, but just follow the guidance on the NIH website. Indirect costs are still allowed, and the most up-to-date information is on the NIH website, or you can contact the NHLBI program staff, and we can route that to our Office of Grants Management for guidance.
Mike, I think we could probably address, is there a requirement for the type of accelerator partner? And I think there's a bit of confusion what the accelerator partner might be providing, the funding, a loan, investment, equity, etc.
So the accelerator partner may be the entity that provides you with that non-federal funding. However, their main role is to help you develop as an entrepreneur or connect you with entrepreneurs, connect you with follow-on funders. They often have courses or classes that they provide or speaker series on how to improve your entrepreneurial training and understanding. That's the type of role of the accelerator partner. There is no specific type that we require. However, we do identify ones that are stronger than others. And certainly, if you apply for the R61, we can help you better understand or scope for them, but we don't recommend any specific accelerator partner. Let's assume that you should be collaborating only with US entities at this point, given some of the things I mentioned earlier with subawardees. So there's some additional questions about letter of intent and how to submit the letter. So as you may be aware, the NIH is consolidating some of their functions, one of which is peer review. So the old instructions for a letter of support to the NHLBI, you may still follow them. However, review from now on will be done at the Center for Scientific Review, centrally located within the NIH Office of the Director. And so we are trying to work with them to update some of these NOFOs that have requests for a letter of intent. They are not required, but they just help the scientific review officers identify reviewers.
There's another question you may have responded before, but it's still here. For R61 portion, can it be split between startup and sub-awardee if that knowledge is housed within the sub-awardee? For example, gene therapy vector design is best done with a nonprofit CRO, but then their startup would take it through studies and clinical experiments.
Right. So again, there are no requirements on where the work should be proposed to do, other than it has to be a domestic entity go until further notice. And really, the lens should be where can you get it done with the data that's appropriate for future FDA scrutiny and review if you're at that stage of development. And then I saw some questions about Payline. This is not a program that has a Payline. We fund similarly to other programs like the SBIR Phase 2, where we have a zone of consideration. Generally, priority scores between 10 and 40 fall in that zone. So that gives us programmatic flexibility to identify projects that are of high impact and excitement, but may have not reviewed as well. And then generally speaking, we are funding about 15 to 20% of the projects. The preclinical program is set up very differently. So, you may propose a series of studies, and then because it's through a service contract with the coordinating center, we may renegotiate the actual studies for budget or other reasons. So there is no similar pay line for the preclinical programs, or even a zone really, because we don't score these the same way through, or they're not reviewed the same way. And then the last question I'm seeing from 1255, well, it was the last question. Let's see, so there was something about the data on the awardees. And again, if you go to the Catalyze website, you can get all the data that's publicly available about the awardees so far. And blood disorders question, those should be routed to Nitin because he is in the blood division, and he and his colleagues there would be able to address the project most effectively. And a $2 million quote from a CDMO. Again, the guidance that we provide on the website is generally speaking, we fund in the 1 million plus or minus range. However, you should contact either the coordinating center or the NHLBI and discuss your project. We may know of different ways to support it, either in pieces or through other funding mechanisms.
Fabulous. Okay, 2 minutes. Maybe we can take 1 or 2 more questions. Let's see. Does a letter of intent go to both the Catalyze POs or the CSR?
So for the time being, letters of intent should be directed to the email address within the NOFOs, which is the NHLBI Chief of Scientific Review. I think that's what the full descriptor is.
Great. Sudhi, any additional questions that you'd like to finish up with?
No, nothing.
Perfect. All right. I think that's all we have time for today. Anybody that has asked a question in the chat or in the Q&A that we did not have a chance to respond to, or those that I've reached out to for your email address, we'll get those questions answered via email in the next coming days. So keep a lookout for those responses from us. And as always, please visit our website, nhlbicatalyze.org. And you can reach us via email at catalyze_info@rti.org. So thank you very much, everyone, for joining. And thanks so much, Mike and Diana, for your presentations.